Let's get started. The countdown continues. 5 lectures counting today. We're almost there. Yes? Okay. Last time I got through quite a bit of translation. And in fact, I got through all the translation that we'll be talking about with respect to prokaryotes with one exception, and that's translation. So I want to talk about, not translation, termination. Okay. So termination, I want to say a brief word about, and then we will move on to talk just a little bit, very briefly, about eukaryotic translation, which, as you'll see, is not that different from prokaryotic. Okay, let's see. So I was right here. I want to just say a word, remind you that translocation, which is the part where the elongation occurs, it requires another G protein. I mentioned that last time. And the G protein that it requires is called EFG. And EFG works sort of like this. EFG sort of fits in and sort of squeezes everything over. And I mentioned that on that translocation step that we saw everything sort of slide down. As the ribosome is doing translation, it must move down that messenger RNA three nucleotides at a time. And that movement down the RNA is catalyzed by and is the responsibility of this G protein called EFG. And the EFG cleaves GTP as an energy source in facilitating this entire process. After that translocation has happened, then this G protein, which has the GDP in it, goes away. And we're left with an open A site for the next transfer RNA to come in and continue the translation process. So the main things I want to talk about here actually are the termination. So termination of the translation process has to occur, and termination of the process occurs when one of 3 specific codons appear in the A site. So when we look at the genetic code, and I encourage you to go back and look at that table, you will see that there are 3 codons that are called stop codons. And stop codons means literally that. When one of those stop codons appears in the A site, translation stops. And now I'm going to tell you how that process occurs. Okay? So we saw in the initiation of translation that there were proteins called initiation factors, or IFs, that started the process. We saw during the elongation process that there were protein factors called elongation factors that, or EFs, that continue the process. In the termination phase, we see proteins called termination factors, and they're actually called RFs. Okay? RFs. There's RF1, RF2, and RF stands for release factor. So what happens during termination, as I said, is we see the appearance of one of the stop codons. So there are 3 possible stop codons: UAA, UAG, UGA. And yes, I think those are worthwhile knowing. I think it's also worthwhile knowing what the start codon is, which is AUG. That's UAA, UAG, and UGA. Those 3 will stop translation when one of those appears in the A site. Now, what happens is when one of those appears in the A site, there's no transfer RNA that will form a stable base pair with it. No transfer RNA that will form a stable base pair with it. So they come in, they get kicked out. They come in, they get kicked out. They come in, they get kicked out. Well, if that went on forever, of course, nothing would happen. There's a release factor, an RF, that comes in and that can, in fact, form base pairs with the stop codons. There are 2 different release factors in E. coli. In eukaryotic cells, there's 1. You don't need to know the numbers or any of that, but just know that there are release factors that come in. And what they bring in is very interesting. So they're actually carrying something to the place where translation would occur. And it's something I haven't told you about so far, and now I want to tell you what it is. If you think back when I talked about EFTU, During elongation, I said EFTU was carrying the transfer RNA so that it could be fitting in the ribosome so that translation could occur. A peptide bond could be formed, etc. And I said that EFTU was the most abundant protein in E. coli by a long way. So it's a very important protein. And one of the things I've shown you is that it actually grabs onto the transfer RNA, and it grabs onto the transfer RNA in such a way that it covers up the bond between the amino acid and the transfer RNA. It's covering it. It's physically covering it and preventing water from being able to attack it. That bond between the transfer RNA and the amino acid is very unstable in water. So as soon as it gets put on, there darn sure better be enough EFTU to come and glom onto there and protect it before water has a chance to break that bond. So EFTU is not only carrying that transfer RNA to the ribosome for translation, it is also protecting that transfer RNA from losing its amino acid due to water. When everything gets set up in the ribosome and the EFTU goes away, that bond is in a position in the ribosome such that water can't get to it. And again, you have a stable bond that's there as long as there's no water. Well, you begin to see where this is headed now, because the release factor, what it does is it carries a single molecule of water to the ribosome. And that molecule then cleaves the bond between that long polypeptide chain and that transfer RNA. When that happens, the long polypeptide chain goes floating away. It leaves the ribosome. And there are other proteins that come in, particularly one called RRF, which stands for ribosome release factor, which helps to at least partially take apart that ribosome. Now, why do you suppose it might be beneficial to just partially take apart that ribosome? What's that? Right, because on a prokaryotic system, we've got multiple genes. So if we completely take everything apart, then we've got to completely make it again. But if we only partially take it apart, then it's possible for the next gene to start translation faster, and there's less effort that's needed in making that whole thing happen. So that's what happens in prokaryotic translation. Okay. Questions about that? Okay. Yeah, Jenny?
Are these release factors sort of physically similar to the tRNAs?
The question is, are the release factors physically similar to the tRNAs? And yes, a portion of them is similar. I say are, but a portion of them is, in fact, similar to the tRNA, which is how it fits in. It actually physically fits into the A site. So it has to, yeah. Yes? So the water is breaking the bond between the transfer RNA. Remember, we have a transfer RNA in the P site, and it's what's attached to that long polypeptide chain that's out there. So it's breaking the bond between the transfer RNA and that long polypeptide chain that's there. That polypeptide chain is now released as a fully functional protein. Okay. So that's prokaryotic translation for you in a nutshell. Let's say a little bit about eukaryotic translation, and the main things I'll talk about eukaryotic translation are where there are differences. So first of all, you remember that eukaryotic translation has bigger ribosomes, okay? Bigger ribosomes, bigger subunits. The overall process occurs mechanically in just about the same way. Great similarities to it. The differences in eukaryotic translation relate to things that are unique to eukaryotic systems. So, for example, eukaryotes make messenger RNAs, each of which only contain a single gene. And as I mentioned when I talked about the Shine-Delgarno sequence, eukaryotes usually translate starting at the first AUG. So there isn't as much concern for locating where that is. The ribosome slides along, it finds it, and it stops there and waits for everything to get started. Okay? Eukaryotic translation includes initiation factors like we saw in prokaryotes. It includes initiator tRNAs that have methionine. Notice that it's methionine and not F-metmethionine. That is the formulated methionine. We don't have that. And that's because the eukaryotic ribosome actually protects that methionine and keeps it from doing the side reactions that can happen in prokaryotes if we don't put that formyl group on there. So we have just a regular methionine in here. We assemble it very much the same way. The small subunit comes in first. We have the initiation components. We have the large subunit. And at the end of that initiation phase, just like in prokaryotes, we have the initiator tRNA, that methionine tRNA, in the P site. Remember that the very first one comes into the P site. All the other ones come in to the A site. Okay? Now, elongation, termination, again, I'm not going to go through the details because they're very, very similar to what happens in prokaryotes. The proteins that are involved have different names. And again, I don't think the names are particularly relevant for us or important for us, so we're not going to worry about the names. But I will show you something that's significantly different. And this actually relates to two structures that we see on the eukaryotic messenger RNA that we don't see on the prokaryotic messenger RNA. You may remember when I talked about eukaryotic messenger RNAs, I said that they had two features that were interesting. One was a cap at the 5' end, and the other was at the 3' end, a sequence I called poly A. The cap is actually recognized by the initiation factors that help to start translation in eukaryotes. So the cap is a sort of a flag that this is a messenger RNA and this is something that you're going to translate. Okay? The other thing that happens is that there are factors in this complex getting everything started. This EIF, that stands for eukaryotic initiation factor. Don't worry about the names. As I said, that's not important. But factors in this complex getting everything started also recognize the 3' end of the messenger RNA. I'm sorry, I've got it over here. The 3' end of the messenger RNA. Now, so here's a protein binding the cap. Here's proteins binding the 3' end. And what we see happens in eukaryotic messenger RNA is we start the process over here. It continues. It comes over here. It hits a stop codon. Everything comes apart. Now, the beauty is that this is a circle. Okay? And the apparent importance of the circle is that when the ribosome comes apart after it hits the stop codon over here, it doesn't have to travel very far to get over here to find the start codon. If the tail is just hanging off and it's not in close proximity, then when this guy releases, it could go anywhere in the cell. But because it's happening over here, it really has to traverse a very short distance before it can start translation of another copy of that same protein. So the circle kind of keeps everything corralled up, if you want to think about it that way, so that the ribosome doesn't stray very far and it continues doing this translation process. Okay. Let's see. That's what I want to say about. That's what I want to say about the eukaryotic process in general. There are a couple of other things we have to think about eukaryotic translation that we don't have to think about in prokaryotes, and that relates to organelles. Because remember that prokaryotes don't have organelles. They don't have endoplasmic reticulum. They don't have lysosomes. They don't have nucleus. They don't have mitochondria. And those organelles have to have specific ways of getting specific proteins. We talked very briefly about that last term when we talked about eye cell disease. If you recall, that was a disease that involved migration of proteins from the Golgi apparatus to the lysosomes, and that was screwed up in eye cell disease. Now we're going to see a little bit about how translation has to be considered when we have proteins that have to get into specific organelles. Okay? So remember that translation occurs out in the cytoplasm. Translation starts in the cytoplasm for a certain class of proteins, but it doesn't end there. And that's what I'm going to show you right now. So for proteins, there's a picture of, actually it's kind of a cool picture. There's a picture of the endoplasmic reticulum, and you can see these little dots all over it. Okay, you see the dots? Those dots are ribosomes, and they're translating. So for a certain class of proteins, those proteins start being translated in the cytoplasm, and then they migrate to the endoplasmic reticulum. The endoplasmic reticulum is the place where proteins that are going to be processed, whether they're modified or they're targeted to different organelles, that's where they all accumulate. And this is how they accumulate, because they're being synthesized and the little protein is being squirted into the inside of the endoplasmic reticulum, as I'll show you in just a second. Okay? So this process I'm getting ready to describe is only for proteins that have a specific destination besides the cytoplasm. Cytoplasmic proteins don't do this. Cytoplasmic proteins they start their translation in the cytoplasm and they finish their translation in the cytoplasm. They have no place else to go. Proteins that have places to go have to go through the endoplasmic reticulum. Okay. Well, how does the cell know to get them there? And the way the cell knows to get them there is by use of a very interesting thing called a signal sequence. Okay? A signal sequence. A signal sequence is, there are several signal sequences that are shown here. The signal sequence is near the amino terminus of the protein. And as you can see, it's not a very specific sequence. There's quite a bit of variation in what those signal sequences are there. But the common thing that they have is that this signal sequence is composed of amino acids that are fairly hydrophobic. So a cluster of hydrophobic amino acids near the amino terminus will target that protein for the endoplasmic reticulum. And I'll show you how that happens in just a second. So these are all proteins that have signal sequences that are going to target them for the endoplasmic reticulum. Okay? All right, so here's how it happens. Oh. Is it you? I'll hold this out here and see if it makes a difference. Okay. How does it happen? Well, it happens as a result of action of something called a signal recognition particle, or an SRP. Don't worry about that last figure I just showed you. It's shown much better here. Okay? What we see happening in this is on the top we've got cytoplasm. On the bottom we've got inside of the endoplasmic reticulum. And as you can see in this figure, translation starts in the upper left in the cytoplasm. It starts, and as translation proceeds, the N-terminus of the polypeptide chain starts coming out that hole in the ribosome. If there is a nonpolar region close to that N-terminus, this protein called the signal recognition particle, or the SRP, grabs a hold of it. So that's what's happened here. The signal recognition particle has recognized in this sequence near the amino terminus that there is, in fact, a nonpolar region, and it's getting ready to drag this whole thing down to the membrane of the endoplasmic reticulum. Okay? That's what's depicted here. So we see at this point that the signal recognition particle has bound to a receptor in the membrane. It's called the SRP receptor. And now the two have bound together, as you can see here. Look at this guy. It's another G protein. Another G protein is involved now in helping this whole thing to happen. GTP is a very, very important energy source in translation, and we see it happening also in the targeting process. Okay, so it docks with this guy here, and once it docks, it lets go of, and also cleaves GTP, it lets go of that amino acid chain that's been made, and it gets squirted into this thing called the translocon. The opening of the translocon allows that polypeptide chain, as it's being synthesized, to be dumped into the inside of the endoplasmic reticulum. So what's happening is this guy is sitting here, and instead of the ribosome moving, the messenger RNA is being moved through it. It's like pulling a thread through something. Okay? So the messenger RNA is being moved through it. This provides translation to happen, and we see ultimately that a stop codon is encountered. When the stop codon is encountered, the protein is released into the endoplasmic reticulum's lumen, that is the inside, and the ribosome comes apart. Now we have a protein that's inside the endoplasmic reticulum. It can be processed. It can be modified if necessary. It can also be prepared for where it ultimately is going to go. And yes, there are tags inside of here about where this protein is destined to go. Tags in the sense of specific amino acid sequences. Okay. Sorting is the way in which things move. So we see that the endoplasmic reticulum can bud off pieces, and those budded off pieces can go assemble and make the Golgi apparatus. The Golgi can then be a traffic cop and say, okay, some of you guys are going over here to be kicked out of the cell. Some of you guys are going to go over here and you're going to sit in the membrane. Some of you guys are going to go over here, you're going to be in the lysosome, et cetera, et cetera, et cetera. So as a result of this, the proteins in a eukaryotic cell get to their target destinations and perform the functions that they need. The process that had the eye cell disease that I talked about is deficient in this process right here. And if there's something wrong with the flagging that says I'm supposed to go to the lysosome, then those target proteins don't get to where they need to be. And of course, the cell has severe consequences as a result of that. Okay, I should ask before I do that, questions on that or anything else I've said here so far? Yes, Megan.
I was just wondering, so the ribosomes are actually floating around in the cytoplasm?
Ribosomes are floating around the cytoplasm, always floating around the cytoplasm unless they're docked. But even they're still sitting physically in the cytoplasm. Laurie?
Going back a little bit to the circular complex of the messenger RNA.
Yeah.
How does it get degraded and stop translation? Its tails are essentially covered up.
I understand the question. How does it get what?
How does the messenger RNA get degraded if its tails?
How does the messenger RNA get degraded? Okay, so the covering up of that doesn't physically cover every single thing that's there. So a nuclease can still come in and start chewing inwards from that 3' end. It doesn't completely protect it from degradation. That degradation does still happen. Kyle?
You said that the signal recognition protein will hold the hydrophobic amino acid chain down to ER. How is it moving? How is SRP moving that?
How is SRP moving that complex down to the endoplasmic reticulum? By the same process we've seen over and over, diffusion. It's a diffusion-driven process. Diffusion-driven process. Diffusion is your friend. Jenny?
Since all these signal sequences are a little bit different, are there more than one signal peptidase that has to find a cleavage, or does it cleave at always the same place, or could it be different?
Yeah, that's a good question. As far as I know, there's only a single peptidase that does that. But it's a very good question, and I don't know if there may be more than one. As far as I know, there's only one. Other questions? Okay, so we're just about done. The last things I want to say about translation actually relate to design of antibiotics. So antibiotics frequently target translation. So if we can find differences in prokaryotic translation compared to eukaryotic translation, we can design a drug that will target those differences and have little or no effect on eukaryotes as a consequence. There are many examples that are out there. This table describes some of them. Streptomycin inhibits initiation. Tetracycline binds the 30S subunit. Stops amino acyl tRNAs from coming and binding. Chloramphenicol is a very efficient one that inhibits the peptidyl transferase. And there's a name I haven't given you. The peptidyl transferase is the name of the activity in the ribosome that makes the peptide bonds. So the peptidyl transferase is actually that ribozyme, remember the 23S, catalyzed the formation of the peptide bonds. That activity is known as the peptidyl transferase. Here's one that's specific for eukaryotes. Okay? This is frequently used in laboratory to see if a process in a eukaryotic cell requires new protein synthesis or not. So if we stop protein synthesis and we see if the process occurs or not, we can tell if a new protein was needed in order to do that process. It's a very useful tool. Yes?
A question about erythromycin.
Uh-huh.
I think my brother has some allergies. About that.
Allergies, there are allergies to a variety of antibiotics out there. Penicillin allergies are not at all uncommon. And those are strictly a function of the immune system. So has nothing really to do with the antibiotic nature per se, but has only to do with the the fact that the immune system recognizes it as a foreign body. And some of the allergies can be very debilitating. Puromycin causes premature chain termination. This was a very cool mechanism. I think I may have a figure of at least the structure of puromycin. Puromycin looks like an amino acyl tRNA. Okay? It looks like an amino acyl tRNA, but it's not. Okay? So this is what we would have in an amino acyl tRNA is we'd have the rest of the sequence down here. That is the rest of the tRNA sequence down here. At the top part, this guy looks very, very similar, and it looks similar enough that the bacterial ribosome actually binds it in the A site. And when it binds it in the A site, this guy becomes covalently attached to the existing polypeptide chain that's in there. So there's a peptide bond that's actually formed, but this guy can't leave. And so it just gums the whole system up. It physically stops translation by having the ribosome use it, but then it can't get rid of it. And it stops. So it's in a sense like a suicide inhibitor. It's not technically a suicide inhibitor, but it's like one. Okay, that's, I think, all I want to say there. Yes. Okay, we are slogging through here. Maybe we should have a quick song. Then we can start a new topic, okay? This is a song I have never sung by myself in front of a class. It's a very difficult song to sing, so I figured it'll probably be even funnier. This is why I really want help from you. You guys know the song Maria from West Side Story? Okay. I hear some laughs, so it's not a funny song. My song is hopefully funny, but Maria is not. The song is called Translation. So, it starts out by saying, Translation. The most intricate thing I ever saw. From 5' to 3', translation, translation. I'm the background singer too here, okay? The final step that we know about the central dogma. You guys can do this part too. Amino, carboxyl, translation, 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 translation. I just learned the steps of translation and all the things they say about tRNA are true. Translation to form peptide bonds in translation. The ribosomal cleft must bind to add FTU. Translation. Oh, wait. AUG binds the F-mets cargo. 16S lines up Shine and Ogarno. Translation. I'll never stop needing translation. The most intricate thing I ever saw. Translation. That was really bad, wasn't it? I've never done that by myself. I've always had people helping me with that one. So, okay, that loosened it up a little bit. So that makes up for the fact I don't have any more jokes. You guys know all my jokes now, so I've got to make fun of myself, I guess, to make you laugh. All right, gene expression. So this is going to be the last sort of the biochemical type talks. The last of the talks that I'll give relate to the immune system and the sensory systems, and although they do relate to biochemistry, they're not what I would call strictly biochemistry. This is sort of still pure biochemistry that I'm talking about. Some would call it molecular biology, which is most of what we've been working with the second half of the term. Okay, so gene expression is a phenomenon that is fascinating. It's a topic that we could teach an entire course on. And I know I've said that many times during the term, but it's true. We could teach an entire course on this. And it's a very, very important thing, both for prokaryotic cells and for eukaryotic cells. Cells are always working on the edge. Okay, they're always working on the edge of Am I responding properly or not responding properly? The consequences of not responding to environmental stimulus or other things is death. So being on that edge, the cell is hopefully always properly responding to the signals that it's getting, to the needs that it has, to the signals it gets from the body, all kinds of different things. And these signals, with respect to gene expression, relate to, ultimately, how much functional protein is made. So gene expression relates to how much functional protein is made. As we will see, there are several mechanisms for controlling that. They include control of transcription. We've already talked about this. I said some promoters are what we call strong, meaning that the cell makes a lot of RNA. Some promoters are weak. The cell makes less RNA. If there's less RNA, there's going to be less protein made. Just a fact of life. We will see that gene expression, we've already seen that gene expression can be affected by splicing. What specific protein is made, how the mixing and matching is done. Splicing, if there are errors, cause problems. So if the splicing is not efficient, we don't get as much functional protein made. We will see differences in translation. I mentioned that if we messed up the Shine-Dogarno sequence, we would see a reduction in the efficiency of translation. That, too, is going to affect how much of a given protein is made. We'll see effects on stability of messenger RNAs. We've talked about that a little bit. In the case of eukaryotes, we saw how the messenger RNA could be degraded either from the 5' end or the 3' end. The 5' end was fairly protected. The 3' end was not. So the length of the poly-A tail really was a factor. And so the stability of the messenger RNA is a factor in terms of how much functional protein we have. And last, though I won't talk about it, another factor is how long a protein is allowed to exist. Cells target proteins for degradation in some cases, in many cases actually. So how long that protein is around and functional really determines how much at any given time is active. And these are only some of the mechanisms, I'm not going to talk about all of them, but these are only some of the mechanisms involved in a cell controlling how much active protein that it has. If it doesn't control this properly, the cell is in danger. So it's very, very important the cell do that. The first thing I'm going to talk about is the most studied gene expression system that I'm aware of, and it occurs in bacteria. And it's relatively simple. It's called the lac operon, LAC. The lac operon is a collection of genes that controls the ability of a bacterial cell to use the sugar lactose. So the lac operon is a collection of genes that allow a bacterial cell to use the sugar lactose. Okay? Notice it's a collection of genes. When I use the term operon, I defined that before. That's a collection of genes, okay, on a single messenger RNA. That collection of genes usually has a common function. In this case, the function is metabolism of lactose. Okay? And so I'm going to say some things about that. Well, let's look at the control region, in the promoter region of the lac operon. One of the things that we see, and of course you haven't memorized the sequence, and I don't expect that you will, but one of the things we see looking at the lac operon is we don't see control sequences that look like Tata boxes very well. And I told you that if we don't match that TAT, AAT very well, what happens to the efficiency of transcription? It goes down. It turns out that the cell, the bacterial cell, has the ability to have lactose transcription turned almost completely off, and in other cases to have it turned on as one of the strongest promoters in the cell. Now, so far, the only thing that you've seen that affected transcription in prokaryotic cells was something called the sigma factor. This system has something else, and I'm going to show you that as well. Okay? So, this is a promoter that binds to different factors under different conditions and allows the bacterium to control how much of this operon is being made. Remember, we've got multiple genes. We have 3 genes in the lac operon. It takes a lot of energy to make a messenger RNA. The cell doesn't want to waste that energy unless it needs that. And one of the places where it would need it is if the cell needs lactose. If it has lactose. So lactose is milk sugar. So if a bacterium falls into a container of milk, it might be very nice for it to be able to make the lac operon. But if it falls into, let's say, a Pepsi Cola, which has no lactose in it, it would be wasting energy if it made the lactose operon. So this is an example about how it has to respond to the environment in which it finds itself. Well, how is this made possible? It turns out it's made possible by the binding of proteins, specific proteins, to specific DNA sequences. And these specific DNA sequences are in the region of the promoter of the lac operon. In the case of the lac operon, this region has a specific name. In addition to being the promoter, it's called the operator, because it helps to operate whether the gene is being made or not made. Well, before I say about how it works, I want to say some general things about proteins that bind to DNA molecules. These things I'm going to describe to you happen in both prokaryotic and eukaryotic cells. And what we see when we look at proteins that bind to DNA molecules is we discover that they have several structural similarities if we compare different proteins. And those similarities relate to functional regions of these proteins. And a functional region, I hope you remember from the last term, is called a domain. So a domain of a protein that binds to DNA, what we're doing is we're comparing domains here. Okay? So one domain that we see in DNA binding proteins is something called a helix-turn-helix. I think I briefly mentioned it last time. A helix-turn-helix looks something like this. Helix, a turn, and a helix. It's pretty straightforward, right? Another class let's see, that's not, okay. So in fact, these are all helix-turn-helix examples. Okay, you can't quite see it here. It goes back behind there. The helix-turn-helix wraps around here. Helix-turn-helix over here. And these are examples of different proteins that bind to DNA sequences that have that feature. So helix-turn-helix is a common feature we find in DNA binding proteins. There's also a thing called beta strands that can actually physically insert themselves into a DNA molecule. And you can see these beta strands here. Remember, the beta strand was a structure that we see inside of many proteins. The beta strands have physically inserted themselves into the DNA double helix. Now, they're not only just inserting themselves, they're inserting themselves usually in specific places. The specific places correspond to specific sequences. So they're recognizing and binding to specific sequences, all the things that I'm talking about here. A common structure we see in eukaryotic cells, the other ones I saw we talked about are both prokaryotic and eukaryotic. A thing that we see in eukaryotic cells is something called a homeodomain. And it looks like this guy right here. Again, these are just names I'm throwing out at you. It's not something that we're going to draw or recognize or anything like that. And the last one is a very cool and interesting one called a leucine zipper. And I always like to tell the story about how people discovered leucine zippers. Leucine zippers can do a couple of things. One, some of them can bind to regions of DNA, such as you see here. Others can interact between either two different proteins. So these might be two strands within the same protein. These might be two different proteins. And one of the ways that people discover leucine zippers, where they have all these amino acid sequences of a whole bunch of different proteins, and one of the things you get when you get sequences, you say, gee, I wonder what these sequences do. And so you start looking for sort of unusual or interesting or funky things. And when they discovered the leucine zippers, the funky thing that they saw was that they had several proteins that they knew bound to DNA and or interacted with each other. And what they saw was that these guys had every 7 residues, there was a leucine. Every 7 amino acids, this guy had a leucine. And you look at this and you figure, well, that can't just be my randomness that's there. If it were randomness, it wouldn't be occurring every 7. It must have some specific function. And it turns out it does. Every 7 residues, you're seeing interaction between leucine, 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 leucine. Leucines are hydrophobic amino acids. Their side chains like to interact with each other, and what we make is a structure that looks very much like a zipper. It's kind of cool. Okay? So leucine zippers have a leucine residue every 7 amino acids, the side chains of which interact with each other, and it peels apart very much like a zipper does. Okay, zinc fingers we also see as DNA binding structures. As their name implies, they include an atom of zinc in that process. Not surprisingly, zinc is a positively charged ion, and DNA is a negatively charged structure, so the zinc can help facilitate interactions between the two. And that's basically it, what I want to say for binding proteins. What I want to do now is talk a little bit about the metabolism of lactose, and then I'm going to talk about how the lac operon is controlled. Okay? So lactose, you remember from last term, is a disaccharide. Or even if you don't remember it, it's still a disaccharide. All right? It's a disaccharide made of 1 molecule of galactose and 1 molecule of glucose. It's called milk sugar because it's found in milk. And to break it down requires an enzyme known as a beta-galactosidase. Okay? At least in bacteria. We have an enzyme in our bodies called lactase that does the same thing. Okay? But in bacteria, E. coli in particular, they have an enzyme called beta-galactosidase. And what it does is it cleaves that galactose and glucose, splits the bond between them to give us free glucose and free galactose. When we talked about glycolysis last term, we talked about a way that galactose can be made into glucose. So we see that all of the components can be used in glycolysis if this bond can be broken. If this bond cannot be broken, then you don't have free glucose, and this guy just sits there and does nothing. So the bacterial cell wants to turn on enzymes that include this one, and this is the most important of the enzymes, wants to turn on enzymes that include this one when lactose is present, and it doesn't want to have this going on when lactose is absent. Okay? So if it falls into a Pepsi Cola, it's got other problems besides lactose. It might get, I don't know, diabetes or something. Okay. Well, biochemists are lazy people. I've told you that before, and I've given you numerous pieces of evidence, including my own lectures. And in the case of this particular enzyme, beta-galactosidase, biochemists like to have easy ways of studying it. And the easy way that they have of studying it is like we talked about last term. Remember last term we had the serine protease, chymotrypsin, that we had the colored intermediate, that when the bond got broken, we made a yellow color, and all we had to do to see how much chymotrypsin we had was to see how much yellow that was produced. Well, the similar thing happens with this. X-gal is a molecule that's made in a laboratory. It's an artificial substrate that works with beta-galactosidase. That is, beta-galactosidase will catalyze a reaction on it and release a molecule that spontaneously gives a blue color. The spontaneous part you don't need to worry about. All you have to worry about is the fact that if I take this artificial chemical X-gal and I add it to beta-galactosidase, I will get blue color, and the more beta-galactosidase I have, the more blue color I will get. So it's very similar to the situation with the chymotrypsin from last term. This is very nice because I can add this to cells, and cells will take it up just like it's galactose. Just like it's lactose. They'll take it up, and they start making blue color. You make blue cells. If and only if they're making beta-galactosidase. If they're not making beta-galactosidase, or they're making a very tiny amount of beta-galactosidase, you'll get no color or very, very, very faint blue. Okay, so this is an important consideration for us in our study of this operon. Okay. Now, if I have a batch of E. coli cells and they're growing, let's say, on glucose, and let's say the glucose is even starting to get a little bit low, they're getting a little bit hungry, if I measure how much beta-galactosidase they're making before I give them any lactose, what I discover is they're not making very much, if any, at all. In other words, essentially no blue color. When I add lactose, what I see within the span, look at the time frame here. This is about 15 minutes up to about 60 minutes. It's about 2 generations. Within 2 generations, I'm up to a very high level of beta-galactosidase that's been induced by the lactose. So lactose and lactose alone has caused the cell to go from making no beta-galactosidase to making a lot of beta-galactosidase. So we know, therefore, that lactose has to play a role in turning this process on. Okay. Here's what the lac operon looks like. So now we're looking at the DNA that contains the operon. The lac operon has several features. The genes are called Z, Y, and A. And the only one that we're really going to focus on much is Z. That's the beta-galactosidase. One of these genes is called a permease, which simply opens up the cell a little bit more and lets more lactose in, but it can get in without that. Okay? And another one is involved in making a byproduct of lactose that turns out to be important, but again, I'm not even going to name the enzyme for you. So we're going to mainly focus on this guy right here. The operon starts here and moves to the right. Okay? Here are 2 regions called P and O. Okay? O is the operator, and P they're using here to technically call promoter, although the reality is the operator is part of the promoter. All right? But they distinguish it promoter, operon. So in their scheme, operon is between the promoter and the start site of transcription. Okay? I'm going to tell you what the operator does in just a second, but I want you to keep that in mind. Promoter, operator, start site of transcription. What's this guy over here? All right? This guy is not a part of the operon. When the operon is turned on, it has nothing to do with this guy right here, at least in terms of turning this guy on. So when I turn the operon on, I'll make a lot of this, but I'm not going to affect this. But I plays a very important role in the process, and it's a protein called the lac repressor. The lac repressor. The lac repressor is a DNA binding protein that, when it's made, binds to the operator, and it prevents RNA polymerase from being able to start transcription. This is a very effective way to completely turn off transcription of a specific gene. Remember how I said we've got controls, we turn the volume up, we turn the volume down? Here's an on-off switch. If the lac repressor is sitting on this operator, this guy is off. Nothing's going to happen. Okay, everybody with me? So, that's kind of cool. Well, we can see how we can turn that system off. Here's the actual structure of the lac repressor. We see how we can actually turn that transcription off. How in the world, then, do we turn it on? Okay? Well, the way we turn it on is with lactose, if you remember the experiment that I just described to you. It turns out that the lac repressor has a binding site for lactose. And when the lac repressor binds to lactose, the repressor can no longer bind the operator. Very cool. RNA polymerase can come in and do its thing. There's still another piece of the puzzle, though. What's the piece that's missing? Remember I said that the promoter wasn't a very strong promoter? Okay? So it turns out, I'm not going to talk about it today, I'll talk about it next time, but there is another protein that comes along and plays a role in making that promoter very attractive. It's not a sigma factor, it's another DNA binding protein that I'll talk about. Okay? I think that's a good place to stop for today. Let's do that, and I will see you on Friday.